So now we're going to look at one of the metabolic pathways of glycolysis, right? So when we think about glycolysis, what is the first thing you can say? You can see that glycolysis is the lysis means breakdown of glyco, which is sugar, right? And the breakdown of sugar happens throughout our bodies, different microorganisms, and the final product of glycolysis is a generation of ATP. So if we want to generate ATP, we're able to utilize ATP for energy, and then we actually make a lot of physiological phenomena take place, right? So if we go over the actual pathway of glycolysis, it begins off with the metabolite glucose, right? Hence the word glyco, right? So glucose gets phosphorylated into glucose 6-phosphate by an enzyme called hexokinase. Hexokinase is prevalent everywhere. There's another form of kinase where it's called glucokinase, which is prevalent in the liver, right? And it utilizes ATP, so adenosine triphosphate, and transfers one of the phosphate groups onto the glucose molecule. So now you have this G6P, glucose 6-phosphate. Another important factor about this first step, this is one rate determining step, meaning this is irreversible, right? So as soon as glucose gets converted to glucose 6-phosphate, it does not revert back. All right. Next, you see an isomerization. So isomerization means that the chemical maintains its molecular formula, however, the bonding changes. So glucose 6-phosphate isomerizes into fructose 6-phosphate, right? And you can notice that here, this is a reversible reaction. This can go back and forth, whether or not, right? So now we have fructose 6-phosphate, okay? Fructose 6-phosphate gets phosphorylated again into this next molecule here called fructose 1,6-phosphate. So this 1,6 or this 6 and this other 6 that you see, this is the region of the carbon that the phosphorylation is taking place. That's why they're labeled based off of these numbers. Okay. Another thing is really important too about this phosphofructokinase here. This is another rate determining step, meaning it is irreversible. It utilizes ATP again. So the ATP, the phosphate from adenosine triphosphate, gets transferred over to the fructose 6-phosphate in order to have this new molecule. All right. Now, fructose 1,6-phosphates goes through this pathway. There's this enzyme called aldolase. Aldolase breaks it up into DHP, so dihydroxyacetone phosphate, or G3P, different from this G. This is glyceraldehyde 3-phosphate, right? And between these two molecules, again, another example of here, this is all reversible. This can go back and forth. So the only ones that are so far that I've taught you that are completely irreversible is this hexokinase and this phosphofructokinase. Everything else is reversible. And now this DHP and this G3P gets reorganized back together with this triose phosphate isomerase, right? So they have the same chemical integrity, right? So the chemical molecular formula, but however, the final product that's supposed to be in order to continue this glycolysis pathway is G3P, glyceraldehyde 3-phosphate. All right. So now we get to another location. Again, another reversible location in this pathway. G3P, glyceraldehyde 3-phosphate, gets converted into 1,3-biphosphoglycerate. Okay, this is a very important metabolic enzyme during hypoxia. Whenever the red blood cells are present and they are beginning to become denatured or dying, this chemical gets released, and which allows for proper changes in hemoglobin affinity. So here we have this cofactor coming in, NAD+, right? NAD+ serves as a specific metabolite in order to remove a hydrogen from this G3P, right? So this G3P becomes oxidized, right? So it loses a hydrogen. And also you can see here from this diagram, that you also have another phosphate group. So all of this is done from this enzyme called G3P dehydrogenase. So every time you see a dehydrogenase, you're removing a hydrogen group, right? Specifically here, you remove actually two. So 2 hydrogen groups are actually removed. So you'll have a free floating hydrogen and another hydrogen that has reduced the NAD+, right? And now here you have the 1,3-biphosphoglycerate. So 1,3-biphosphoglycerate here, you have this conversion, so this removal now, again, requiring another kinase, of phosphoglycerate kinase here. And this phosphoglycerate kinase converts this 1,3-phosphoglyceride into 3-phosphoglyceride. So you remove one of the phosphate groups, and that removed phosphate groups is reattached back to the ATP, right? So ADP then now gets converted into ADP. So now back to this step here, you have 3-phosphoglycerate. It needs to get converted to 2-phosphoglycerate. The way it does it is to phosphoglycerase mutase. This mutase also is very similar to isomerase, right? So it maintains its chemical molecular formula. However, the bonding order changes, right? So you see the phosphate group goes from the 3-carbon to the 2-carbon, all right? And next you have a dehydrogenation reaction where the enolization takes place. So you have this enolase enzyme where it removes this water from this 2-phosphoglycerate, all right, in order to have phosphoenol pyruvate. Now we're at the last chemical reaction before we have our final product. So this whole mechanism is to generate ATP and also pyruvate in order to enter the Krebs cycle. So final step is another rate determining step. So it's irreversible here. So you have 3 of them again. So you have, I'll make these very obvious. Here. Okay, so these are very important irreversible rate determining step where the phosphoenol pyruvate gets converted into pyruvate, the final product that we're looking for, right? And this is by, again, having the ATP coming in and ATP production, because the ADP comes in and removes this phosphate group, right? And so if you look at this whole diagram here, you can notice that beginning with the starting product of glucose, you end up with pyruvate, 2 ATP molecules, and an NADH. All right? And so all of this is created in order to prepare the body for the next cycle, which is going to be the Krebs cycle and the oxidative phosphorylation.